they're including the first step, or the stage 2 as part of this Krebs cycle. Okay, so we have the pyruvate coming in from glycolysis. It's a 3-carbon molecule. It becomes a 2-carbon molecule of acetyl coenzyme A, giving off an NADH and a CO2. Now, that 2-carbon acetyl coenzyme A enters this cycle, and this cycle is called Krebs cycle. The cycle starts with a 4-carbon molecule. The 4-carbon molecule is called oxaloacetate, which is also known as OAA. Okay, so when you take a 4-carbon molecule and add a 2-carbon molecule to it, then you're going to end up with a 6-carbon molecule. Okay, and this 6-carbon molecule is what we call citrate. Okay, actually they do have it there. Okay, right there, which is why this is called the citric acid cycle. Citrate, or citric acid, is the first stable product of this cycle. Okay, that's why we call it citric acid. Okay, the first step is just a rearrangement step. You don't need to really worry about it. You're never going to have to know the names of these intermediates. The only ones you have to know is this one, oxaloacetate and citrate. Okay, but watch what happens. When we go from this 6-carbon molecule, we go down to a 5-carbon molecule. So we're going to lose a CO2 molecule here. We then go from a 5-carbon to a 4-carbon molecule. So we're going to lose another CO2 here. Okay. So if you think about it, we lost one CO2 here when we went from pyruvate to acetyl-CoA. Those two acetyls came in, those 2 carbons came in, then we lost one and we lost 2. Okay. So by the time we finish all of Krebs cycle, all of the carbohydrate that was in the original glucose molecule has now been converted to CO2. We have completely oxidized the sugar by the end of Krebs cycle. Okay? This reaction happens twice because there are 2 pyruvates coming in. Okay? So we've oxidized all the carbons in the sugar. But we also have to think about the electron carriers because that's the point of this particular step. Okay. We had one oxidation occur here, so we had to have an NADH produced here. We go from 6 carbons to 6 carbons, nothing happens. 6 carbons to 5, that's an oxidation step. Since it's an oxidation step, we have to have something to come along and grab those electrons. Okay? What do we normally have come and grab electrons for redox reactions in biology? Right. NAD is going to come. So what you're actually going to get is a formation of one NADH here when you go from 6 carbons to 5 carbons. Okay? Because we lose another CO2 here going from 5 to 4, we also produce another NADH at this point. Okay, something interesting happens here. We get a series of chemical reactions occurring between this 4-carbon molecule and this rearrangement, and this step causes a substrate level phosphorylation event to occur. So we actually generate 1 molecule of ATP per turn of Krebs. Okay. We then have a whole series of rearrangement steps to get us back to oxaloacetate. Well, this step here, from this 4-carbon molecule after the generation of ATP to this 4-carbon molecule, there is another oxidation step, but we change carriers. Okay, and the reason is that the energy in the bonds of this 4-carbon molecule are insufficient to transfer electrons to NAD+. There's not enough energy in this 4-carbon intermediate to cause the reduction of NAD to NADH. However, there is enough energy in this 4-carbon molecule to allow for the reduction of NAD2 plus to NADH. So we change electron carriers at this step, just at this one step. We then have another molecular rearrangement, and then one final oxidation step occurs here, and again we generate another NADH. Okay, so starting from pyruvate, how many NADHs will we make for one single turn of Krebs cycle? You forgot one. There's an NADH that comes up here, right? From 3 carbon to 2 carbon, right, is an oxidation step. So there's 1, 2, 3, 4 NADHs. Okay, 1 ATP and 1 FADH2. But because it's a glucose molecule, this happens twice. Okay, so we reduce electron carriers. So total amount of NADH produced is 8. Total amount of FADH2 is 2. And total amount of ATP is 2. Okay? So remember your numbers. Okay, so what's going on? We've fully oxidized glucose at this point. Okay? We started with C6H12O6. Okay? We've oxidized that completely to CO2 at the end of Krebs cycle. In the process, we ended up with 4 ATPs. Okay? Where did we generate those 4 ATPs? Starting with glucose, where do we generate all of our ATPs up to this point? 2 from what step and 2 from what step? In glycolysis, we get 2, and in Krebs cycle, we get 2. That gives us a total of 4. Okay? So what's the point? Because that's not a hell of a lot of ATP. What did we produce lots and lots of instead of ATP? The NADH. The electron carriers are the key. Okay? The electron carriers are hydrogen carriers. Okay? So Krebs cycle, including pyruvate oxidation, produces a large number of electron carriers. We produce 8 NADHs and 2 FADH2s. So 10 different electron carriers. Okay. What we're going to do then is we're going to take these electrons from these electron carriers and take them to the electron transport chain. The electron transport chain is how we're going to generate ATP. Okay. When we looked at secondary active transport, what provided the power to move sucrose into the cell? Hydrogen ions. Okay. Now, in this situation, we're going to use the same concept, hydrogen ion gradients, to generate ATP using an enzyme. Okay. In that secondary active transport system, how did we generate the electrochemical gradient? Right, we used a primary active protein pump. The problem in this case is we can't use an ATP pump to create this hydrogen ion gradient, because then you'd be using ATP to make ATP, which makes no sense. It'd be a one-to-one loss. So we have to use something else. What we're going to do is we're going to use the electrons in the carriers to go through a series of redox reactions. And we're going to use the energy of the redox reactions to pump hydrogens across. That's going to produce our electrochemical gradient to allow the formation of ATP. Okay? That's what we are basically doing. Okay? Why are the electron carriers so important? Okay? Because the electron carriers are temporarily holding the power, the reducing power of glucose in its bonds. Okay, so at this point, let's do some energy accounting. When we take a pyruvate to carbon dioxide, okay, we're going to produce 3 carbon dioxides, right? Because you start with 3 pyruvate, sorry, 3 carbon molecule, you have to end up with 3 carbons. So that's 1 to 1. In the process, we're going to take 4 NADs, convert them to 4 NADHs. We're going to take 1 FAD, convert that into an FADH2. We're also going to generate 1 ATP through substrate level phosphorylation. Okay, so we've got money stored in the bank as these 2 piggy banks, and we've generated 1 ATP that the cell can use on its own. The net gain of this process in Krebs is 2 ATP. Okay, 8 NADH2s and 2 FADH2s. Okay, so if the yield is only 2 ATP, how is Krebs an adaptation? It's the value of the electron carriers. Okay, because with these electron carriers, we've now got the ability to move electrons and hydrogen ions that we need to make the ATP. Okay, so what we're about to do is we're going to break open this piggy bank, and we're going to cash out the electrons in these 2 piggy banks. Okay, so that's what we're going to do in Krebs. Okay. So the summary, and then what Kirsten loves so much, which is a Mr. W song.
Aerobic respiration is the most important process used to produce energy for a cell. It involves several pathways, including glycolysis, pyruvate oxidation, the citric acid cycle, and electron transport. Various sugars, fats, and proteins enter these pathways, and they can all be broken down to produce ATP energy for a cell. In this animation, we will focus on the citric acid cycle, also called the tricarboxylic acid, TCA cycle, or Krebs cycle. The citric acid cycle takes place in the matrix, or fluid, of the mitochondrion. This is where mitochondrial DNA is found and where fatty acid breakdown takes place. The citric acid cycle involves 8 chemical reactions that use acetyl-CoA and oxaloacetate to produce carbon dioxide, NADH, ATP, and FADH2. The NADH and FADH2 are electron carriers that can be used in the electron transport chain to make more ATP for a cell. Let's take a look at the major steps of the citric acid cycle. In the first step of the cycle, a 2-carbon molecule and a 4-carbon molecule are combined to form a 6-carbon molecule. That 6-carbon molecule undergoes multiple biochemical changes during the cycle, and at the end, the original 4-carbon molecule is produced. Each time a carbon molecule loses 1 carbon, a carbon dioxide is released. So 2 carbon dioxides are formed during the conversion of the 6-carbon molecule back to the 4-carbon molecule. So where does the 2-carbon molecule come from to begin the citric acid cycle? This molecule, acetyl-CoA, is made from pyruvate. Pyruvate is a product of glycolysis, the other major ATP-producing cycle in the cell. Pyruvate is transported into the mitochondrial matrix, where it is oxidized to acetyl-CoA by pyruvate dehydrogenase. This step produces 1 NADH and 1 carbon dioxide for each of the 2 pyruvate molecules made from glucose. The acetyl-CoAs are the starting point for the citric acid cycle. In the citric acid cycle, the acetyl group from acetyl-CoA is transferred to oxaloacetate to form citrate. 4 different enzymatic reactions then lead to the formation of succinate. These steps produce 2 carbon dioxides, 2 NADHs, and 1 ATP. Succinate is then recycled back to oxaloacetate through 3 more reactions. These steps produce FADH2 and 1 more NADH. All of the energy molecules made in the citric acid cycle along with those produced by glycolysis, are essential for fully functioning cells, as errors in these pathways can lead to life-threatening diseases.
It's a substrate level phosphorylation, Grant. The only place oxidative occurs is in stage 4. Because we're not using the power of electrons to synthesize ATP. We're in the case of Krebs, we're literally transferring a phosphate from a sugar to the ATP. Okay, you'll kind of see that here, I think. Oh, before I start this, okay, you do not, I repeat, you do not need to know the names of the intermediates. He gives them, you don't need to worry about them.
Matrix of the mitochondria of all our cells is the cycle of reactions that won Hans Krebs a Nobel Prize. The cycle takes the energy and food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. And Krebs makes FADH2, its function is the same. Krebs also makes some ATP, another claim to fame. We're talking Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Right before the cycle's a transitional part, links Krebs to glycolysis so Krebs can start. Enzymes rake CO2 off a pyruvate, yields an NADH and acetyl-CoA. Acetyl-CoA carries carbons too, with all the energy that derives from food. It's a highly reduced molecule with energized electrons. It fuels up the Krebs cycle as it cycles on. Now at the start of Krebs, this acetyl-CoA has the 2 carbons it carries in, ripped away. Enzymes hook these on oxaloacetate with carbons 4, make 6 carbon citric acid. Who could ask for more? Notice 3 carboxyl groups on the citric acid. That's why the cycle's also named for tricarboxylic acid, or TCA cycle, if it's acronyms that you prefer. Krebs in honor of its discoverer, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Enzymes work on citric acid and remove the CO2, and other enzymes modify and oxidize it too. The 5-carbon result is alpha-ketoglutarate. Krebs cycle, it's so great. Every oxidation can power the reduction of NAD+, which gains electron-carrying function, become an NADH, that energy sensation, which later on in respiration powers ATP creation. Another oxidation follows another CO2 remove, leaving us with a 4-carbon molecule. Another NADH results from this oxidation, which Krebs does its energy transformations. This 4-carbon molecule, succinyl-CoA, still has lots of energy enzymes can take away. A series of reactions yield 1 ATP, the cell's main energy currency, leaving just enough energy for enzymes to reduce an FADH to FADH2, and 1 less NAD+ will also get reduced as the final electron carrier NADH gets produced. We've harvested what energy came in at Krebs' start. Now we have oxaloacetate at this final part. Oxaloacetate is the commencement and finale, ready to meet acetyl-CoA, and here at the final tally. Krebs goes round and round, such an ancient cycle, spinning like the wheels of my bicycle. Krebs like the axle of aerobic respiration. I breathe out CO2 with every exhalation. We've walked through the cycle, so now let's review. Input is acetyl-CoA with carbons too. The carbons get removed, releasing CO2. Exhaling sends the CO2 out of you. The cycle's functions: energy transformation, 3 NADH, 1 FADH2 creation, and also synthesis of 1 ATP, which cells directly utilize for energy. For every glucose cells absorbed, the cycle runs 2 times. As long as cells get fuel, Krebs is running just fine. It precedes electron transport chain, it follows glycolysis. It's spinning round and round in the mitochondrial matrix, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2.
The key point is to remember the numbers more than anything else. Come on, where is it? Blank 2. Why is it there? It should be on classwork. Okay. Last thing we're going to be looking at then is pyruvate oxidation. Okay. For this one, you guys will need access to a Jamboard as well. Okay. Oh, believe me, there's something else I can replace it with. Much worse. Especially if you know the black-eyed peas. I got a feeling. Okay. So stage 4 is the electron transport chain. This is the payout. This is where we generate the maximum amount of ATP possible. Okay, so up to this point, what we've done is to the end of Krebs. We've got a total of 4 ATPs through substrate level phosphorylation, and then a whole bunch of electron carriers, right? NADs and FADs that are going to carry the electrons to this last stage where we're actually going to produce a massive amount of ATP, roughly 36 moles. Okay. So up to this point, glycolysis for every mole, or sorry, for up to this point, cellular respiration, for every mole of glucose used, we've produced 4 moles of ATP. With this last step, every mole of glucose will end up producing 36 moles of ATP. Okay, so it's a massive change. And the point is to make ATP. Okay, so the ATP accounting up to this point. Glycolysis produced 2 ATPs, substrate level phosphorylation. So did Krebs. Okay, but obviously your cells run on a lot more than 4 ATP, right? Because of this fact. So there's got to be a better way to generate more ATP, and there is. That better way is called the electron transport chain. Okay, it's a series of proteins that are built into that inner mitochondrial membrane, the cristae. Okay, and they're basically transport proteins or permeases and enzymes. Okay, what they're going to do is they're going to transport electrons down this chain, and we're going to link the redox reactions of these electrons, right, to pumping hydrogen. Okay, so this is it's a similar idea to secondary active transport. This will yield 36 ATP from one single glucose, but, and this is the but, the system only works if oxygen is present. No oxygen, the system shuts down. Okay, that's why this is aerobic respiration. You must have oxygen, otherwise it doesn't work. Okay, and it's a system that kind of looks like this. Okay, and we'll be talking about these blobs in a minute. Okay, so just quickly reviewing the mitochondria. You've seen this slide already, so I'm just going to flip through it. You don't need to worry about it. Okay, what I'm going to do now is I'm going to take a closer look at this section of the cristae. All right, so if you look at this slide, okay, we've got this light blue line here representing the cristae. This area up here then is your inner membrane space. This area down here is your matrix. Okay, so the inner mitochondrial membrane is there. intermembrane space, mitochondrial matrix. Okay, the three enzymes here are number 1, NADH dehydrogenase, number 3, cytochrome BC complex, and number 4, cytochrome C oxidase complex. Okay, those are the names given these 3 blobs. There's also 2 circles here. These circles are cytochrome Q and cytochrome C. Okay, I don't care if you can if you can spell these correctly, but you should know, the light blue line is the cristae. It's the inner membrane of the mitochondria. This light blue line up here is the outer membrane. Cytoplasm will be out here. Okay, so cytoplasm, outer membrane, inner membrane space, cristae, matrix. And then we've blown up this section of the cristae. I don't care if you spell these right or not, okay? But you should know this is NADH dehydrogenase, this is Q, this is BC complex, and this is C oxidase, okay? So those names you should know. Okay. So the electron carriers that we've been talking about, FADH and NADH, okay? Glycolysis produced 2, Krebs cycle produced 10, okay? What we're going to do now is we're going to break open these piggy banks and we're going to start making ATP. I'm going to do this in 2 parts, okay? So the first part, I am going to show you the electron transport chain part first. After that, I'm going to give you the generation of ATP. Okay. Now, this is Q4B. Electron transport chain. I am actually going to need to run this over 2 pages, so the Jamboard will consist of 2 pages. Okay, I'm going to start on page 1. First thing I'm going to do, so I'm going to make, this is going to represent my mitochondria, okay? And the first thing I'm doing, okay, it's in black, is I am making the cristae. So this is going to represent my cristae. 1, 2, 3, 4, 1, 2, 3, and 4. Okay, so this is my cristae. I will have 3 blobs in this in a minute. Okay, so up here is my inner membrane space. Down here is the matrix. This is the 2 lines represent the cristae. Okay, blob number 1, which is located here, and the shape of the blob doesn't really matter. Okay, that's my first protein complex. And that, in blue, is NADH dehydrogenase. Okay. My second blob, which I believe is going to be in green, it's going to be here. And that should be cytochrome. Where's my cytochrome BC complex? Okay. So that's the one in green. I would actually suggest that you draw this along with me while I do it, but it's up to you. You could just, if you want, grab it from Classroom when I put it up there later. The third complex, which I will do in yellow, is going to be here. And that third complex is cytochrome C oxidase complex. Okay, so those are my three carriers, or my three protein complexes that go through the membrane. I also have two other complexes. They're more like movable molecules. One sitting there and one sitting up here. Okay. This one is called Q, and this one is called C. Okay, so this is my electron transport chain. Okay, so if it's an electron transport chain, it's going to need a source of electrons. Okay, ultimately the source is going to be food, right? Glucose. But what's going to, based on the name of this particular protein, what's going to immediately provide electrons to this electron transport chain? Yeah, NADH. So, excuse me, I'm going to have an NADH coming in here. Okay, that NADH molecule, okay, could be coming from glycolysis. It could be coming in from pyruvate oxidation. It could be coming in from Krebs cycle. It doesn't really matter where it's coming from. You just have to know that there is a source of NADH coming into the electron transport chain. This molecule, NADH, is going to convert back into a molecule of NAD plus. Okay. So when NADH becomes NAD plus, what chemical reaction occurred? Oxidation or reduction? Okay, it got oxidized, just lost its electrons. Something has to take those electrons. What literally happens is two electrons go from here onto here. Okay, so we get two electrons that get transferred there. Okay, now because this is an electron transport chain, in order for this NADH dehydrogenase to accept electrons from NADH, does it have to be more electronegative or less electronegative than NADH? Okay, and that's true of the entire chain, because the electrons are going to get passed on from here to the next component. So Q has to be more electronegative than NAD dehydrogenase. BC complex has to be more electronegative than Q, and so on and so on. So the electrons are going to continue to move. They're going to pass on to here. Okay. Then from here, they're going to pass on to here. Okay, so we'll have two electrons here. They will then move from here to here. Then from here, they're going to move on to here. Okay. Now, remember, this is the same electrons that originally started in NADH. It's not two different electrons. It's the electrons that originally started in NADH have moved along and been transferred along this chain. Okay. Now, if the two electrons can't get off of the last complex, what's going to happen to this pathway? Yeah, it has to stop. If this pathway stops, okay, can another NADH come and dump more electrons? No. So this chain would stop, which means the NADs from Krebs can't get rid of their electrons, so Krebs cycle has to stop. Pyruvate oxidation can't get rid of its NADs, so it has to stop. What's the only system left running then? Glycolysis is the only one that's left running. And that's why when you don't have any oxygen, you go into fermentation. Glycolysis, everything else has to shut down because there's no way to move electrons through this chain anymore. Okay, that's why oxygen is so important. So to get these electrons off, we have to use a final electron acceptor, and that final electron acceptor is oxygen. So what's going to happen is you're going to have, not 1.2, half an oxygen molecule and two hydrogen ions that get picked up from the matrix, okay, are going to get converted, are going to pick up the electrons, okay, and what we're going to get instead, what are we going to produce then here? If you take half an oxygen molecule and two hydrogens, what are you going to get? Yeah, you're going to get one molecule of H2O. Okay, so what we've done is we've fully oxidized that NADH to make water, and we've moved electrons through this electron transport chain. The question becomes why? What's the point of doing all of that? Well, remember, what we want to actually create is a hydrogen ion gradient, an electrochemical gradient, which means, I said before, we're going to use the power of these redox reactions to pump hydrogen across the membrane. So here's what's going to actually happen. We're going to take, as electrons go from NAD to Q, you're going to have two hydrogens get pumped across this membrane. They're going to come out on this side. Okay? So every time a pair of electrons, every time a pair of electrons go through this complex, you're going to have two hydrogens get pumped across here. You're going to have another two hydrogens get pumped across here. Okay, and you're also going to get two more hydrogens pumped across here. Okay, this is how we build up a hydrogen gradient. Okay, why did we build up this hydrogen gradient? Okay, if I go over here and build the rest of this, so there's the rest of the cristae. What you're going to have, just sitting a little bit downstream from those permeases, is going to be a structure that looks like this. And the name of this particular structure come on, this thing is an enzyme. Keep doing this. That box off. Called ATP synthase. What do you think the job of ATP synthase is? It makes ATP. And what it literally does is every pair of hydrogen ions that go through this thing okay, so you're going to have two hydrogens because there's an electrochemical gradient, right, that we've created using those electron transport chains. We've pumped hydrogen. There's a high hydrogen concentration over here, low concentration here. Hydrogen is going to come back through this enzyme. Okay, when it does so, ADP plus phosphate gets converted to ATP. Okay, so let's think about the way this works. Every NAD resulted in how many hydrogens being transported across? 6. When those 6 hydrogens come back, how many ATPs can you make? Okay, rule number 1, NADH makes 3 ATPs. Okay. Now, let's look at the other electron carrier. Will FADH2, does it carry the same amount of energy or equivalent amount of energy as NADH? Yes or no? No, it can't. That's why at that one step in Krebs cycle, I said there's not enough energy to reduce NAD plus to NADH, so we change carriers to FAD. So FADH2 has a little less energy than NADH, which means it can't pump 6 electrons or 6 hydrogens across. What happens instead with FAD is pretty similar. With FAD comes in at a slightly different point. So we have FADH2, that becomes down here FAD2 plus. So this goes to that. And the electrons from this thing end up in here. They enter at Q. So we're going to have 2 electrons from FADH2, okay, moving on to Q. That being the case, how many hydrogens can FAD move across the membrane? 4, which means FADH2 makes how many ATPs? 2. Rule number 2, FADH2 makes 2 ATPs. Okay, so in general, NADH2 makes 3 ATP, FADH2 makes 2 ATP. Okay, this system only works if hydrogen can't do what? Because it's easier than saying NADH plus H plus. Yeah, it assumes that the hydrogens can't get through this inner mitochondrial membrane. If the hydrogens could pass through this membrane, would ATP synthase have any function? Yeah, it would short circuit this because you couldn't make any ATP. Okay, so in order for this system to work, the cristae has to be pretty near impermeable to hydrogen ions. Okay, and it almost is perfectly impermeable. It's not 100%, but it's pretty damn close. Okay, I want you to try a math for me. How much ATP then can you make from one glucose molecule, accounting for all the NADH from all 3 stages, all the FADH2s from all 3 stages, plus all the ATP you make directly from substrate level phosphorylation? What's the total maximum amount of ATP that you can make from one single glucose molecule? See if you can figure that out. Remember the rules. NADH makes 3 ATPs, FADH makes 2, plus the ATPs from substrate level phosphorylation. What's the total maximum ATP you could produce? Yeah, it's 38 molecules of ATP, right? This is the massive payout. From 1 mole of glucose, we get 38 moles of ATP. That's huge. That's more than enough to run the cells of a multicellular organism like you. Excuse me. And that's why we do this. This is also why we are so reliant on O2. This is what gets shut down. If someone gets cyanide poisoning, cyanide basically sits on this complex and prevents electrons from moving off onto oxygen. Exactly, Abby. Now, I'm about to screw that up in a minute. Before I do you want me to copy this Jamboard over to Classroom? Do you want access to it? Yes? Okay. Share. Change link to anyone with a link. Copy link. Done. Edit. True, but do you think you only have one of these running at once? Within a mitochondria, do you think you only have one set of electron transport chain? Yeah. So you got thousands, right? In fact, this is kind of neat. This is what the electron transport chain actually looks like. Well, it's an animation of it from Harvard. So there's a mitochondria. It looks like a bacterium. Proteins being shuffled in. There's the electron transport chain. This is ATP synthase. That's cytochrome C. There's a hydrogen pump. This is the neat part. This is ATP synthase. It's a spinning little motor. It's a protein motor. It physically moves like this. Every pair of hydrogen going through causes that thing to spin, and that spinning motion powers the synthesis of ATP. And then you got lots of mitochondria in your cells. Okay, so let's continue on with this. So everything I've just been talking about in that Jamboard, I'm going to reiterate on this. Okay. Oh, sorry, it's replaying. Stop. Minimize. Yeah, okay. So we've talked about this bit. Okay, so going through this one more time, right? We have NADH. That's going to that'll split the hydrogen atom into an electron and a hydrogen ion. Okay, so NADH becomes that. Hydrogens get split. You're going to have electrons move on to the first complex. They then go on through Q to the second complex, through C to the third complex. That's if you're NADH. If you're FADH, you come in slightly later at Q, but otherwise do the same thing. Hydrogen gets split. You move an electron onto the chain, through C, onto the chain, and then finally you have oxygen pick the electrons up and become water. Okay, you're going to basically pee that water out of your body if you don't need to use it. Okay, so the electrons carriers pass electrons and hydrogens to the electron transport chain. The hydrogen is cleaved off the NADs and the FADs. Okay, the electrons then are passed from one electron carrier to the next in the mitochondrial membrane, and those flowing electrons now provide the energy to do work. Okay, the reason that the electrons go down this chain, and these are all the components of that chain that we've been talking about, it's because each component of the electron transport chain is more electronegative than the previous. So the electrons want to keep going down until you get to the most electronegative atom down here, which is or compound, which is oxygen. Okay, so it's a controlled oxidation. Okay, we're controlling the release of energy and we're capturing it as ATP instead of releasing it all at once as heat. So we make ATP instead of making fire. The process of synthesizing ATP from the power of electrons is called chemiosmosis. It assumes that the mitochondrial membrane is impermeable to the hydrogen ion. Okay, so hydrogens get split, the electron, right, and then the hydrogen ions get moved across these membranes. Okay, this builds your proton gradient. But what powers the hydrogen pumps? The movement of the electrons through the electron transport chain. As the electrons go through, hydrogens are going to get pumped across. Same way, depending on NAD or FAD. Okay. Now, before we go on to the next bit, because I want to talk a little bit about why we get variations in the total amount of ATP, because as Patty calculated, we should be theoretically be getting 38 ATPs for every sugar molecule we burn. Turns out that's not quite true. We're going to take a look at why that isn't completely true when we get back from break. Okay, it's 1:30 now. So take, I need to use the washroom also. Take 15 minutes to break. And we'll get back together at 1:45. Okay, see you then. Okay. All right. So in general, the rule is FADH2s make two NADH, ATPs, and NADH2s make three. That's not always 100% true. Okay. So up to this point, what have we done when we say we did it? We set up a hydrogen ion gradient. We allow the protons then to flow back through ATP synthase. We make ATP by combining ADP with phosphate. Okay, so we can make ATP here. This is what we call a proton motive force because it is driven by protons. So chemiosmosis, which is the process by which we make ATP, is the diffusion of ions across a membrane. Okay, so we build up a proton gradient so that hydrogen ions, by using an electron transport chain, so hydrogen ions can flow through ATP synthase to build ATP. What chemiosmosis does is it links the electron transport chain here to ATP synthesis here, right? That's this bit. And that's the point. Okay, so here's what we do. We took pyruvate from cytoplasm and we strip electrons off of it in the form of electron carriers. Those electron carriers from stage 1, 2, and 3 are then passed on to the electron transport chain to provide energy to pump the protons across the across the membrane. Oxygen at the end is joined with those protons to form the water molecule. And the protons that we pumped across are going to diffuse back through ATP synthase down their concentration gradient, which drives the synthesis of ATP. Okay. So this is where the accounting changes a little bit. What about the NADH from glycolysis? Okay. And it turns out, Patty's right, 38 is a theoretical maximum of ATP that you can make, but cells don't always make the maximum 38. Okay. Like pyruvate, the mitochondrial membrane is not permeable to NADH. So here's the cytoplasm out here. Mitochondria is the wall of the mitochondria is this the cristae is this brown line here. Here's NADH from glycolysis. Look what happens to it. It actually passes in most cells inside the mitochondrion to an FADH. Okay. So that means in the majority of cells, the NADH from glycolysis will make how many ATPs? 2 or 3? Only 2. Okay. The exception are heart and brain cells. Okay. They use a different method of getting NADH into the inside of the matrix, and they pass NADH from glycolysis to another NADH inside the mitochondrial matrix. So in heart cells and brain cells, they make the NADHs from glycolysis make 3 ATPs. Okay, so brain and heart make the maximum. They make 38 ATPs. Well, you're going to learn that that's not true either, but you can wait on that till next year. So for now, I'm only partially lying to you. The brain and the heart will make 38 ATPs. But because every other cell in the body only passes the electrons from NAD in glycolysis to FAD, how many ATPs will most other cells make? Yeah, they make 2 less, 36. Okay? So the cells of your muscles, the cells of your liver, of your stomach, your small intestine, your big toe, they make 36 ATPs. Your heart and brain make 38. Why do you think your heart and brain make more? Yeah, they literally need more energy. They need the extra ATP to keep running. Okay? So in most cells, NAD goes to FAD, but in brain and heart, NAD goes to NAD, which is why we get this difference. Okay? So 2 ATPs by substrate level phosphorylation in glycolysis. Okay, 0 to 2 ATP, depending on the shuttle that transports the electrons from NAD into the cytosol. Okay. 2 ATP by substrate level phosphorylation at Krebs. And then 34 ATP roughly by oxidative phosphorylation using these NADs and FADs. Okay. 2 plus 2 plus 32 to 34 gives you 36 to 38 ATPs total. So, summary. Okay, there's your equation, right? And I mean very approximately 40. Okay, where does the glucose come from? These are a series of questions. Okay, our food. Where did the oxygen come from? Atmosphere. Where's the CO2 come from? Yeah, it comes from the carbohydrate. It comes from the food. Where does the carbon dioxide go? Exhaled. Where does the water come from? Does it come from the food? The electron transport chain. Yeah, it's a combination of hydrogen and oxygen. The water is formed out of the oxygen. Okay, it's not. Okay, I'll talk about that in a minute. Where did the ATP come from? Yeah, it basically came from the reduction of the oxidation of the carbohydrates. Okay, what else is produced that is not listed in this equation? Okay, the NADH at the FADH2. Yeah, absolutely. Why do we breathe? Well, specifically, we need the oxygen as a final electron acceptor. Okay. Actually, let me go back one more. So, why is this process a little deceptive? Why is this equation a little deceptive? And then I'll try and answer Emma's confusion. Okay, it is an oversimplification, but the deception is here. Okay, what this equation looks like, okay, is it looks like that carbohydrate gets split. It looks like the carbohydrate is a source of water, and the carbon combines with oxygen to form CO2. Okay, but what's the actual source of water? Is it the carbohydrate? No, the source of water is actually from the oxygen. Okay, that's why this system is a little bit. This is a bit of a deception. It's a bit of a lie. Okay, what's confusing, Emma? These questions or the process? What parts got you confused the most? So you don't get how the glucose becomes pyruvate? Right? I mean, the basic idea, and I'll get back to you in a second on that then. Hang on. Okay. So what's the final electron acceptor in the transport chain? It's oxygen. Okay. And this is only half the unit. We still have photosynthesis to go through. Okay. What happens if oxygen is not available? The electron transport chain is going to back up. Okay. Because probably next week, because we'll finish photosynthesis this week, have a lab. So yeah, it'll be mid next week or. So I have 2 more tests to mark in this class, so I should be able to give them back today at some point. And then I got to start marking the other class. Okay, so if there's no oxygen, the electrons can't flow down. Okay, NADH and FADH can't release their hydrogens. So ATP production stops. The cells then run out of energy, and you die. Right? This is why oxygen is so vital. Okay, because cyanide is going to block this step. Okay. Now I've got a single video. This is the one I was talking about earlier, right? That basically summarizes all of cellular respiration. Oh, it's an ad. Okay, I can play it.
This goes out to all my biology students. If listening to this song helps you better understand and remember the process of cellular respiration, then making it was totally worth it. So sit back, relax, and enjoy the song. Cell respiration. It's a long, complicated process that I need to know for a biotest, but it turns my brain into a huge big mess. Cell respiration. It breaks down food to release energy. Food molecules are glucose specifically. You need oxygen to oxidize thoroughly. Cell respiration. It gives off some carbon dioxide. Also water's made off to the side. But the ATP is why we got on this ride. Cell respiration. It's made up of 3 main stages, but it takes up so many stages. And to know it all seems to take many ages. Glycolysis, glycosamine split, is what's first up in the cytoplasm. Takes 1 glucose, 6 carbons, and splits it up. To do this right, it's phosphorylate from 2 ATP, so 2 ADP are made. Now that the glucose has phosphated, it breaks into 2 molecules that seem like mates to NAD+, coming off the oxidize them, which means they take electrons and hydrogen, and 3 phosphate. Now we're set. 2 ADP, it's really 4, and 3 carbon, met. Phosphates break off, ATP made. Left with pyruvate, there's 2 of them, and they go to the Krebs cycle, which happens in the mitochondria. Oh no, wait, not really, there's transition step, let's count the ATP. 4 were made, but 2 were spent, that means 2 ATP were profited in glycolysis. It didn't need any oxygen to NADH, now have hydrogen, and 2 pyruvate molecules were made in the end. Transition step. And AD plus oxidizes pyruvate. 1 carbon is lost along the way, and both molecules turn into acetyl-CoA. Now 2 carbon, carried by comes A, Krebs cycles next. In the matrix, 4 carbon molecule, already there. The 2 connect. Now 6 carbon gets oxidized by NAD plus. 1 carbon left, CO2 gone. We'll do this twice. Numbers doubled. 4 carbons are left. 1 ATP made, oxidized by the NAD plus and FADH. Molecule change, no carbon lost. Atoms rearranged, so 4 carbons looks like the start. Ooh. And then we'll do it again. Krebs cycle, repeat twice because 2 pyruvates were made in glycolysis. Let's count it up. We now have 4 ATP, 6 CO2, and then these guys 10 NADH, 2 FA, bring electrons to ETP. Electron transport chain, inner membrane, brain of the mitochondria. Proteins in lane. Electrons from NADH pass through 3 proteins, electrons from FADH2 pass through 2 proteins, as the electrons pass through the protein compound. Hydrogen ions called protons too. Inter membrane space, a concentrated place. Protons will return to the ATP synthase makes 32 ATP from returning protons. 6 oxygen molecules get the electrons, and they combine to make 6 water with hydrogen ions. So respiration. Now I know the entire process. If I study, I'll do well on the bio test, and afterwards I'll get a good rest.
Okay, so that's that song. Okay, and it does pretty accurately summarize the numbers for cell respiration. Yes, he's not exactly a perfect pitch singer, but his science is correct. Anyways, I don't know if that would be helpful to put on Classroom or not. Yeah, I think for everyone else it probably will be, because it has all the numbers and it basically summarizes the entire process. So I'm going to put, oops, not a rename, create material, and this is going to be cell respiration black-eyed peas. Topic May 10 to 14. Yeah. Okay. So the summary is there. Do you want more podcast style summaries of a respiration? Either a crash course or an Amoeba Sisters or a Bozeman Science. Okay, so let me look for those and post them in the same uh, link. So YouTube. Crash course cellular respiration. Uh, Okay, there's the Amoeba Sisters one. Okay, so Control C. Add YouTube URL. Add. I'll add a few of them so that ATP, is this one any good? Yeah, as soon as I make it. In fact, tomorrow I'm probably going to make it a Gizmo lab period. Let's see, does he have ATP, glycolysis, pyruvate, anaerobic fermentation, aerobic Krebs? Okay, he does the whole thing. Okay, perfect. So this is the crash course biology one. Yeah, it'll be sometime next week, but probably mid next week at Zoe or later. It kind of depends on how the two labs we have coming up go, basically, and how much time you guys need. So what do we got? Amoeba Sisters and I guess Bozeman is the last one I should use. Control C. Add URL. Control V. Okay, so there are 4 videos. One is a cell respiration song. So I'll put it up on classwork. There, you can see this now. So that's a song there. Okay. That's the Amoeba Sisters. So cell respiration song is this first one here. This is the Amoeba Sisters. This is Crash Course, and this is Bozeman Science. Okay. So you've got different views of this process. Okay. Emma, figure out what it is that you're not understanding, and come and talk to me then over the next couple of days, because this one and the next unit are the 2 big ones that people crash out on in this course because they're the most important units in this course. The other units, like you guys have done okay on cell resp. Most of you are level 3 or level 4, but homeostasis and population dynamics aren't that hard. But these 2 are the ones that blow most people out of the water. So if you're not getting it, you should spend time and focus on this unit and the genetics unit that's coming up. And we are pacing it a lot faster, right? We're covering the entire course in 2 months instead of 5 months like we normally do. So yeah, the pace is a lot faster. Okay, we got about a half an hour. No, we go till 2:20. Okay, so we got about 10 minutes. Let me check out gizmos because I want to see. There's a good cell respiration gizmo that we can do for a lab tomorrow. There should be. Let's see. Cell. But yeah, there is a stem case. Okay, let's see what this is about. Oh, okay, that's one. I remember it. Okay, there's that. The other thing I'm debating about, there should be, let's see if I can find it. Here. Wondering if I sometimes I do a assignment, and I'm just trying to figure out if I should do one for cellular respiration or for photosynthesis. What did I do in the past? Sem one. For you a one. Okay, so did I do it here? Molecular processes, energy and metabolism. Krebs, energy summary, metabolite, fate of food, regulation. Then I did the STEM. Kelvin cycle chem. Okay, that was term one. And in term two, I don't think I did anything. Genetics, population metabolism, STEM, test lab. Okay, so I didn't. So do I have something I should? Do you guys prefer papers or labs? Well, a lab is like a gizmo, right? And a paper would be a research paper. Yeah, it looks like for this one, I don't really like. I could look up a paper, but I think the stem cell, the gizmo is easier. So we'll probably do a gizmo to. Let's see, what do we have tomorrow? Because we still have to get through. Actually, what we have to go through is regulation of cellular respiration and fate of food. I think. Krebs, oxidative, etc. Energy summary, control of respiration. Yeah. Okay. So. Okay, so let me see if I can find that PowerPoint so I can post it for you guys. Forces, 4UR, metabolic processes. There it is. So that can be deleted. Yeah, tomorrow's lectures, in fact, I could probably do it today, but I'd rather not. It's pretty quick once it loads. Okay, turn it into a Google slide. There we go. We should, yeah. The last two units I tend to do just big, massive projects because of the time frame for testing is too um, low. So I tend to do like one project that covers all three expectations. So it's a big project instead of a unit test or a lab. But you will have a unit test and a lab or gizmo for the first 3 units. Okay, carbohydrates, fat. Yeah, so I don't need any of those. So let's delete those slides. Yeah, tomorrow's lecture is really short. It's only 11 slides. Okay, so that's basically it. I will post tomorrow's thing, power slide deck later today. I just have to modify it because I just converted it from a PowerPoint and it looks horrible. So I'll fix that up, post that, and then we'll do a gizmo. Okay, so that'll be tomorrow. And probably make the gizmo due for Friday. Okay? Remember those of you that asked for extensions on your membrane model? So we still have photosynthesis. That's going to take a couple of days, 2-3 days. So we'll probably finish the lecture part of photosynthesis this week, maybe Monday at the latest, do another lab. So we're probably looking at Thursday next week or Friday for the test. Thereabouts. I mean, remember that mid-quad. Well, actually, Monday I can't test you guys because Monday is a BFF day, so it's a day for you guys to catch up. So Monday's a catch-up day for anything that you guys want to redo. The 17th. 19th is mid-quad. All right, so we're halfway through the semester. We're basically a week from halfway through. So we are a little behind where we should be because we should be well into the 3rd unit. The 19th is mid-quad. BFF day is the 17th. 20th is when the mid-quad marks go to parents. Okay. So that means this test probably won't be on your mid-quad mark. And then you got a long weekend after that. And then 1, 2, 3, 4 more weeks and we're done. Because I'm going to try to get the entire course done by the 18th of June. That gives me the 21st and the 22nd to get your marks in. Actually, I have to get the marks for you guys in by the 22nd. So I'm going to be a little pressed for time. All right, it's 2:19. So you guys are dismissed unless you guys have questions and you want to hang out for a bit. That's fine. Otherwise, I'll see you tomorrow. Membrane assignment, I'll get to mark when people start handing them in. The big problem with membrane is that everyone's asked for extensions on it. I've got extensions going to next Tuesday. All right, so I won't get a chance to start marking them till then. Test, I should be getting back to you later today. This class. The other class, I haven't even started marking tests yet. No, you guys are, or hang on, which group is this? Okay, no, this is group B. So the other class is I'll start marking yours probably tomorrow. All right, have a good one, guys.
Mr. Ora,
Yeah.
So I told you this, I think last week, but so May 20th, I have like
Yeah, you
I
have a
can't
thing.
attend the whole